Welcome to the final presentation of Neuroscience Technology class. This is SunghyunKang and GyuHuh. Today, we will be going to present about the introduction of the given thesis and explain more deeply about the imaging of microscopic brain structure. So let's begin the introduction. From the beginning of Kajaw's neuron doctrine, it laid the foundation not only for the modern neuroscience, but also for the modern neurophotonics. Nowadays, there are various research groups related to it, and one of the most famous ones is the Brain Initiative. Here, long debate has been done whether which methodology should be implemented for the specific observations. Thus, they concluded that every material is necessary, and this paper aims to introduce those methods. Note that this paper focuses on methodology for animals rather than humans. Now let me introduce about the imaging of microscopic brain structure. To begin with, fluorescent proteins that originally derived from marine organisms are widely used in labeling such as brain tissue. They utilize the genes that are related to fluorescence and inject them into target domain. Hence, you need to consider the factors related to it. Not only for that, it is necessary to consider other factors such as color or structural proteins to make it FP optimized. Hence, there can be some limitations related to it, and FP cannot be applied for the type of specimen that cannot accept the gene. For tissue clearing, classic protocols usually included a dehydrogenation step using ethanol. However, this step can only be applied to thin brain sections and causes uneven shrinkage of the specimen. Thus, the transition to organic solvents such as the BABB protocol has been introduced to solve the sample thickness problem. But it still has some limitations, and aqueous solutions has arisen as one of the alternatives. Also, the emergence of clarity with light sheet microscopy allowed high throughput analysis with less compromising the specimen. Here is the diagram about clarity. Note that it has different embedding procedure. For super-resolution microscopy, it enabled us to overcome the diffraction barrier of light microscopy. Although there are various types of super-resolution microscopy, it is commonly based on the principle of manipulating on and off the state of fluorescent labels.
The next imaging technique for brain structure is expansion microscopy. It literally expands the size of the specimen. This is used when the diffraction limits the resolution of conventional light microscopy. This follows four work steps. First, infuse and synthesize water-soluble hydrogel throughout the specimen. Then, chemically link biomolecules of interest to the hydrogel and homogenization of the specimen enzymatically or via heat denaturation. Then finally, the physical expansion of the specimen occurs in pure water. The expansion microscopy is used in several areas. First, when studying synapses of the number of the organisms. Next, when tracing neurons in large volumes. And third, to interrogate neurological disease such as Parkinson's disease or dementia. Next imaging techniques are about label-free structural imaging. Label-free means non-invasive and non-toxic alternative fluorescent microscopy. The first label-free structural imaging is MAGIC, mildly autofluorescence imaging by glycerol-induced contrast enhancement. It modifies the sample to increase the autofluorescence signal of the structure. It can serve 3D connections analysis of the brain. The other label-free structural imaging techniques are Raman scattering and CARS to determine the molecular composition of this tissue. SHG for detecting well-organized structure, THG for distinguishing the interface between different compartments, and 3D PLI for enabling the high-resolution analysis of myelinated fiber and so reconstruct the 3D organization of whole brain fiber. The final imaging technique is optical coherence tomography, also known as OCT. OCT is also a kind of Ravel-free structural imaging, but it uses low coherence interferometry to provide a depth-resolved image. In short, it uses different refraction coefficients between white matter and gray matter. Within the gray matter area, optical coherence microscopy has revealed individual myelonated axons and neuronal cell bodies. SOCS with OCT and tissue slicer is the new tool for fundamental research into anatomical and connectional architecture of the brain. It relies on conventional or polarization-sensitive OCT of a volume within the field of view, translating the sample laterally to image neighboring volumes, and removing a layer of tissue using the slicer to exposure different regions for imaging. Then, repeat these steps until the entire block is imaged. This technique is used to distinguish white and gray matter compared with diffusion MRI.
Here are the summary of imaging techniques that are introduced in this presentation. And here are the references. Thank you for your attendance.